Morning. Probably I need to introduce myself, not Professor Jules. Everyone of you know him. Okay. He's going to continue his talk on assembly. Well, when I saw the title, I could only think of around the world in 30 days. But his travel to India has even minimized the 30 days. Okay. Several kilometers and miles he has traveled to be with us. But now it is for him. Please.
Thank you. Good morning. Hope you had a good sleep and are fresh again after yesterday. Just a practical thing. So you remember yesterday with the lab practical that we transferred the colonies to the plate, right, for the assay. And then there was, if you look at the program, we were going to put the fungi, so Rhizoctonia and Sclerotium tomorrow, but tomorrow is a convocation day. I know you also have some other obligations. So I've been thinking about that, and what we can do today is to, shall I use this microphone? It's better, huh? It's working? Yeah, it's working. So to transfer the fungi this afternoon, and then what we will do is we will incubate at a lower temperature so that the fungi, they don't grow so fast. Because if they grow too fast, the assay is not working so well, or at least it will look different. So that's what we'll do then, and then I think we will incubate at 20 degrees. Normally it's 25 or so lower, and then you don't have to come back tomorrow, and also the team here does not have to, do some work here tomorrow, okay? So then your mind is free, and you can, organize your time as you wish, okay? I think today with the practicals, and I'm looking at Guillermo and Victor, I don't think we will go all the way up to 5:30, so there's also going to be a bit more time for you today to work on your literature review. So just, that, we don't want to, push you too much. So you have time to think about the papers, because they are quite, dense, I should say. That's the good word, dense. Have you already started working on the paper? Yeah? How is it going? Yeah? Okay. It's quite interesting. I think I will share that with you today. So on the 20th of February, there was a paper that came out in Nature, and it's super related to the course here. It's about high throughput culturomics. So not so much, amplicon metagenomics, but culturomics. So they developed a high throughput method, computerized, also with machine learning, to collect microbes from a given sample in a high throughput manner, including phenotyping, and some of the trees that we showed yesterday, they are also in there, and they validated that method using, I think, a gut sample from human microbiome, so that they could. Anyway, I will share that with you, not to overload you again, but just, the course is so much focused on, sequencing and stuff, and this is the other end, which is super interesting when you do validation, right, of all the data that you get. Good, so with that said, today I'm going to talk again about plant microbiome assembly. It's the second part. So yesterday we talked a lot about the underlying mechanisms. We did not talk so much about the impact of the plant genotype on microbiome assembly. And that is one of the major parts today. You can imagine that plant genotype will have an impact on exudation, on root architecture, right, on leaf architecture. So in that sense, of course, it will also impact on assembly. Around the world, because, and I will show you the next slide. And this is work in progress. So it may feel a little awkward, this lecture, because several things I'm going to show, it's from work that we are also involved with, which is not finished. And about halfway the lecture, I will ask for your input. Some ideas and what I should do with the experiments. So I hope you can give me some advice, okay? It's good. So we will have sort of like an interactive type lecture where, you know, questions, answers, and then we see how far we will get, okay? But this is a picture that one of my PhD students took in the Andes Mountains, which is in South America. And you can see here some small-scale potato fields, because the Andes region in South America, that's the center of origin of a number of solanaceous plant species like tomato and potato. And this is where we do some of our field work. Yeah, and I will explain you, I will go through the work that we do in this region, but also in other regions, where we look at the impact of plant domestication on microbiome assembly. Okay, and the reason why we look at plant domestication, and this is, I think, a bit of a simplified world map, where you have the centers of food production, the centers of origin. So where do the plants originate from that we nowadays use as a crop for food production, right? And they are in this color. I'm colorblind, so I think it's green, I don't know. But you can see here the Andes region where we work, and also in other places like Mexico, but also in Africa, you have a number of centers of origin of the wild relatives of our current crop species. I was also looking here at India. So I think the map, I'm not so sure. I'm also looking now at Kumar. So I'm sure in India there are also wild relatives of plants that are used, yeah? So I think, you know, this is sort of like a super crude map, which is not fine-tuned to that level. Then, of course, here in China. But I think the most important element of this map is that the centers of food production, so where the crops are produced, are disconnected from the centers of origin, as you can see here. So the most productive areas of a given crop are not in the centers of origin, but they are elsewhere, yeah? And the reason why we look at this is we were interested in what is the impact of plant domestication, so going from wild relatives all the way up to the, you know, improved varieties that we use in agriculture. How has that impacted on microbiome assembly? So have we lost somewhere in that process, which is thousands of years many times, have we lost microbes from the centers of origin, from the wild relatives, that actually have a beneficial effect on plants? And, you know, if we know that, can we bring those microbes back on our crops to, you know, perform the beneficial activities that we have lost? That's the simple concept of this project. And that is called the missing microbes, and there's a number of review papers that have been written on this topic, which are listed there. And I'm gonna make a confession here. So we started with a project, and I will show those slides later, on common bean. So common bean originates from Mesoamerica, so from Mexico here. And we started with a very small project, with a PhD project, actually not in Mexico, but in Colombia, which is not the center of origin, but is a center of diversification of common bean. And we were just 3 of us. So that was Juan Perez, which was the PhD student on the project, and Rodrigo Mendez, a former postdoc of me. And we were, you know, sitting there in Colombia, sampling soils from the native region and also the wild relatives. We went to CIAT, collected the germplasm of common bean and did a number of experiments. And I still remember we were in the mountains, you know, in these native areas, and we were in the farmhouse of Juan's brother. And in the evening we were sitting there, dark sky, lots of stars, you know, it was super fantastic. We were having a beer together. And then we were sort of like, telling each other, how fantastic this project was. Anyway, we were sort of like carried away. And then I still remember Rodrigo said, well, Jos, I think this is going to be bigger than we think it is. And since then, so we developed a website. You can look it up on internet. It's a wiki site. It's called Back to the Roots, right? Where you go. I mean, it's a little movie with some South American music, and it's fun to watch. But that was the very early start of the work that we did on plant domestication. And then we sort of got lost. Well, let's say the concept extended also to other research groups, other collaborations. So we started also to work on tomato and potato. Also here in Ecuador, in the south of Ecuador, close to Peru, to the Andes Mountains, which is the center of origin. And then also a project which I will talk about in my keynote lecture next week, which is on sorghum. I think sorghum is a familiar crop here as well. And that originates from Africa here, Ethiopia, southern Sudan. Right, so we have a project working on that, which is funded by the Bill Gates Foundation. And then a former PhD postdoc in my group, Chung-Soo Song, she is now in China. She started her own research group, and she also connected working on millet. So, so the crops are all different, the plant species. There's a story to every plant species, but the concept, again, is the same. Did we lose microbes during domestication that may have beneficial functions? Now, the concept of the missing microbes is not sort of like a concept that we made up. It was already coined earlier by a medical doctor, microbiologist, Martin Blazer. And there is a nice book here. It's called The Missing Microbes. And the title here says how the overuse of antibiotics and also the changes of diet that we have gone through, how that impact on our gut microbiome and the composition of the microbes that are there. And there's a beautiful cartoon here in a Nature Review immunology paper that they have written there, where, so this is basically the ideal situation where, from the mother to the child, there is a vertical transmission of microbiota. And also in a community context, there's horizontal transmission of microbiota from one person to the other. And that sort of like brings, like a quite diverse microbiota, gut microbiota here, which has a nice impact on innate and adaptive immunity of the human system. So that, altogether leads to a mature adaptive immune response. Are the chairs in front of the screen or not? Okay, so and then the idea is that through generations, and for example also cesarean births, that the vertical transmission of microbiota has decreased, and also there is, giving more the individualistic society, there's also less of a horizontal transmission, so you get a depleted microbiota. So that's, again, that's the concept, that's the idea, which then has also an impact on your innate and adaptive immunity, leading to an increased incidence of autoimmune or inflammatory diseases. Right, so that's a bit of the context in the human microbiome. So also there's a number of groups working on, can we bring back some of those microbes that either through antibiotic use or changes of diet have been depleted, and to bring back to increase their impact on our immunity, right? Okay, so how does that work for plants? So this is a quiz. This is just to show you in terms of what the impact is of plant domestication on very simple things like the phenotype. So the quiz is what, so this is the wild relative of which crop? Sorry? You have to speak up, I can't hear you. I hear wheat. So this is corn, okay? So the second one. Eggplant, excellent, very good. The bottom one. Yeah, it's in the same, but this is cabbage actually. So, yeah. So, and there's, if you go to this page, Genetic Literacy Project, there's a number of, these types of quizzes that you can do. But again, it's to illustrate that during domestication and improvement and breeding, of course, these plant species, they look so different than their wild relatives. So that's one, there's a huge change in phenotype. And then if you go, so again, it was the selection and modification and adoption of wild plants, to our needs. And there was a huge change in seed size, dispersion, and growth patterns. But very importantly, there was also a reduction in genetic diversity. And that has been shown for a number of crop species when you compare them to their wild relatives. And this has led this phenomenon here, so the reduction of genetic diversity, they call this the domestication syndrome. Okay? And that's why, for example, people working in plant breeding, they usually go back to their wild relatives to cross back resistance to specific diseases, right? Because they were lost through the domestication and the improvement and breeding process. Because those processes were actually meant, for example, to get rid of the bitter substances in these and to have a higher productive crop, okay? Now, what is interesting, and so you can look at the impact of plant domestication on microbiome assembly from a plant genetics perspective, and that's what we do. But there is more to the story. Keep in mind, production areas and centers of origin. So what you do when you grow the crops, they are also grown in a totally different habitat, a different environment. So you remember yesterday when I showed you the transact in Europe, going from all the way up north of Sweden to Victor's birthplace in southern Spain, right? So basically that's what we are dealing with. So you change the habitat, and not only habitat, but in the agricultural soils, of course, there's also a totally different management practices. You use fertilizers, you use pesticides to control insects or other diseases. So it's when you study domestication and the impact on microbiome assembly, it's not one specific factor in the plant genetics. You look at many more things. And that's depicted in this. So you have here habitat expansion and altered management. So, and that's listed here. And then here, of course, you have the changes in plant genetics and phenotype. So, and again, coming back to what I explained yesterday about assembly, that has also impact on root architecture, also above ground, of course, and also altered root exudation. Now, we put this in the paper that we published several years ago, and then when you start to look into literature about the knowledge that is there in terms of root exudation differences between wild relatives and their, the domesticated parts, there's very little literature, at least I could find. So there's still a big gap there. Anyway, I'll come back to that later, but because you need it to understand how plant genetics had an impact on microbiome assembly. Okay, so also above ground, and I want to put this slide in here also for people working on phylosphere, insects. There is a super nice paper in 2015 by Chen et al. In the Annual Review of Entomology, where they also looked at the impact, so the domestication syndrome on insect resistance. And of course, needless to say, the habitat, so these are the wild relatives, this is the domesticated crop, maize, and then, the plant density, but also, the habitat has changed, And then, of course, the differences in traits. So this is actually the picture I showed you for the very first time, how the phenotype changes. Then there is quite some interesting literature on the impact of domestication on symbiotic microbes. So the nitrogen-fixing bacteria and the arbuscular mycorrhizal fungi that provide phosphate for the plant. Really, and the bottom line here of several of these studies is that on the domesticated plants you get less mycorrhizal diversity and a reduced responsiveness of the plant to mycorrhizal colonization. And interesting here for the rhizobia, there's even more recent papers. I will show that also. On the domesticated plant species, you get less rhizobial diversity, and the plants that basically recruit these nitrogen-fixing rhizobia, they are less picky about it. So that there is what they call less sanctioning of the plant to rhizobia for their efficacy to provide nitrogen. Because in the new habitat of the production area, there is usually enough nitrogen that is supplied through the fertilizer, right? But for many other microbes, there is very little knowledge about the impact of domestication. For symbiotic ones, there is more literature, but there's still a lot to learn. This is a super interesting paper by Ricardo Soldan here in Communications Biology, where he actually gave two or three scenarios of how would domestication impact on your microbiome composition. And he compared a natural ecosystem here, which we just looked at, and an agricultural system here, where of course you have input from humans on, basically the growth of the plant, the genotype, and also the management practices. But what I found interesting from this paper is the three possible outcomes of domestication that are listed here. So the first one, this is the missing microbe stuff, so you will have a species loss, right? So wild here and the domesticated, you see there's much less diversity here. So the level of selection here, the relative importance, that's the RI, of the domesticated plant is much less than for the wild. Okay. Then a second possible outcome would be that you don't get a change in diversity, but you basically get a replacement. So the composition of your wild plants is different than the composition of your domesticated ones, but the diversity as such is still roughly in the same level. And then, this is also an interesting, you can also may have a species gain. So you may have more diverse microbes on your domesticated plant than in the wild one. And I find that last one also super interesting, because think about it, when you go through domestication and improvement and breeding, you usually in these processes, you grow these plants in soil, and then they are screened in a real life situation, and they usually then pick out the ones that perform best. And basically, the plant breeders usually don't take into account microbes, but maybe unintentionally, they have selected for domesticated plant species that already were able to better recruit microbes that support their growth, right? So that's missing microbes, that sounds very, appealing, but maybe, anyway. So this is where we are at the moment. And there's a lot of work now ongoing in my group, but also many other groups that actually, try to test, this sort of like outcomes. Where are we? So this is what I said. So this is a super nice paper by Stephanie Porter and Joel Sachs in Trends in Ecology and Evolution. Again, so there is quite some things known about the symbiotic microbes, so mycorrhizal fungi, rhizobia. Again, for the other microbes, little. And then last year, together with a colleague in Amsterdam, we wrote sort of like a perspective, sort of like a research roadmap on how to investigate the impact of plant domestication on microbiome assembly. So I'll walk you through this scheme. So again, here, the experiment is that you go to the centers of origin and you collect the native soils from which, you know, your plants recruit microbes. And you also take soils from the production area. And then both soils are then grown to either a wild relative or a domesticated, you know, crop species of that wild relative. So that's one thing. The other thing is that you look at the, you know, the relative importance of the microbiome of your native soils on the growth of, you know, the domesticated or the wild relative by doing soil transplants, right? And then here you can also start introducing biotic or abiotic stress factors, right, to see how that impacts on the phenotype. On the plant side, we proposed that it's important to identify plant genetic loci that are important for the recruitment of these beneficial microbes. Because if you know that, you know, you can also use that knowledge to breed plants, not so much for a specific resistance traits, but for the recruitment or, you know, activation of specific beneficial microbes. And then, of course, once you know this, here you can also look at the, basically the chemistry that is underneath. And that goes back to the, you know, what I just explained about the root exudates. Can we pinpoint specific compounds in root exudates between wild relatives and domesticated plants that are important for recruitment and activation of these microbes? Okay, so let's go back to this map. So this is basically the research program I'm involved with at the moment. And let's go to an example. So the first example that we started, that's the bean. Okay? So common bean domestication, there's quite some nice literature on that. So it originated here in Mexico, and then through the domestication trajectory, the bean traveled, you know, here, northern Peru, Ecuador, Colombia as a center of diversification, and even more south. And that led to two gene pools, what they call the Mesoamerican gene pool and the Andean gene pool. And to do these experiments, what Juan did, and again, that was at the very early days, that's already quite some years ago, we went to CIAT, which is one of the institutes where they keep germplasm of a large number of plant species and also bean. And we collected from that collection, so there was a huge selection of germplasm we could choose from, and we ended up with, actually in retrospect, quite a small selection, but at least at that time it was what it was. So we had a couple of wild relatives, some land races, which are in between, you know, the wild and the domesticated ones, and then a number of, I think we had 4 modern bean genotypes. So that was the starting point of the germplasm. And then what we did is in Colombia, we went to the mountain area where we collected native soils where wild relatives of beans are growing. And we also took soils from agricultural areas in Colombia as the sort of like soils from the centers of production, right? So that was still there. So the soils were collected, the bean seeds here were surface sterilized and pre-germinated, and then there was this experiment where we did basically a common garden experiment, how they call it, where you grow all these bean lines in the 2 soils for a number of days. We synchronized the development for all those ones. That's also a tricky point because the development of the wild relatives and the domesticated is different. So yeah, you can synchronize based on so many weeks of growth, or you can synchronize based on developmental stage, what we did in this experiment, because developmental stage has an impact on root exudation and etc. So that's the way we normalized, and then the roots of these plants were collected, DNA was isolated, and we sequenced the V3, V4 region, which you also have, you know, in the practicals. Okay, and a long story short, this was actually the result. which was published quite a number of years ago already. And what came out of this study, so here when we compare the ancestral bean lines with the modern ones, is that in the modern ones we find a much higher abundance of the actinobacteria and also some proteobacteria here, whereas in the ancestral bean lines we predominantly found Bacteroidetes. So that was the differential abundance analysis that we did and which you also went through in the practicals, okay? Now what Juan did also in this study, he did phenotypic profiling of all these bean lines and he specifically looked at root architecture. And what we noticed is that the wild beans, they had much finer roots and a much more elaborate root density than the domesticated ones. And he did several statistical analyses where he could actually connect, or link, associate the abundance of the Bacteroidetes with specific root length and the actinobacteria with root density. So that was one of the first studies where we actually associated, that is the word, so it's not a causal relationship, we did not demonstrate that, that we could associate those phenotypes with the plant microbiome. And then I think I'm also looking at Victor now. So, the principles of core microbiome, or I think, Vic, you will also cover that in the lectures, right? And also, yeah, and also in the practicals with the core genome, right? Okay, good. So that's what we did here, and that's depicted here. So this is actually, so the core microbiome is the taxa. You can have many different criteria there, but the criterion that we did is that they are present in all the bean lines, in all the soils that we tested. So you can find them back in all of them. Some programs, they use a cutoff that, for example, they are present in 50% of your samples. We set it at 100%, so they have to be present there. And then you can produce these types of maps, right? So this is the core. And what we then did is you can go for each genera, you can go to, these are the bean lines, and this is the relative abundance here, and then you can map them to the native soils or the agricultural soils, right? So you can then see which ones, were more abundant on an ancestral line in the native or the agricultural soil. There's too much detail here to go through, but this is just an example how you can then profile, which microbes were depleted going from wild to modern and depleted going from native to agricultural soil, right? So that's the trick here. And then you can also produce these Venn diagrams, which we did, and then you can see that at the genus level, there is quite some overlap between, for example, agricultural and native soils, but there are some genera that are uniquely present here in the native soils or in the agricultural soils. Think again about the three concepts, right? So what we saw here is that, there was even more diversity in the agricultural soil as compared to the native soil. There is a very simple explanation for this. We did physical chemical profiling of the native soils, and the native soils in the Andes Mountains or in several regions where we sampled here, also in Colombia, they are super acidic, and there is high levels of aluminum toxicity. And that by itself already has an impact on the, the pool of microbes that the plants can select from, right? So, and then when you look at those unique genera here, they are listed here and they are listed here, and you look at the read count and the relative abundance here, it's actually super low. So a next step, for example, and we never came to that point, is to isolate these microbes that are unique for the native soils, for example, and then test them on the wild relative, but also on the domesticated plant, and look for how do they impact on the phenotype, on growth, on resistance to a disease, right? So that's the type of experiments you can do with this type of information. targeting these types of microbes. Some of them are easy, but some of them are, I should say, a pain in the neck. Yeah. So okay, so that's the Mr. Bean story. Okay. We're going to move to the part where I need your input. Okay. It's work in progress. It's a project that we started 2 years ago, a PhD project. So I need your help. And that's on potato. Okay, so this is a bit of the diversity of the potatoes that you can find, you know, among the land races and in the local areas in South America. Yeah, so here again, the journey of the potato, the diversity here on the phylogenetic tree, and of course this is the phenotype of the plant. Basically a bit of a similar story than the bean, although potato genetically is super complex with diploids, tetraploids. It's not easy, and this is again sort of like a journey starting over here, moving northwards and southwards, you know, leading to genotypes that can have a long day length. Yeah, instead of a short day length, it's and more adapted to cold, more adapted to other types of soil conditions. And at some point, of course, potatoes also ended up in Europe here. And the Netherlands now, for example, is one of the biggest producers of seed potatoes worldwide. So, but you know, we are actually totally disconnected from actually the center of origin, right? Okay. So again, here is the map. So the tetraploids, the diploids, and you can simply look at the phenotype of the potatoes. I mean, it's just amazing. And again, here, the phylogenetic tree, it shows you domestication, improvement, and then the breeding step, okay? Leading to where we are now in this type of potato lines. Okay, so going back to the native soils, right? So this is how the region, this is Ecuador, shown here in the map in South America. And what Dario Ramirez, he's the PhD student working on this project with colleagues at the University of Quito, University of Utrecht, and then my group. What Dario did is from the north all the way up to the south of Ecuador, across the mountain range, and I'm not sure about the distance, but I think it's quite a distance. Yeah, let's put it like that. Here is the capital, Quito, and on 14 different sites, and they look like this. So here you have a little potato field here. So he collected soils from this field, which is actually classified as an agricultural soil, and he also collected soils from this region here, which is basically the native soils, which has been, yeah? So for every site, one all the way down, he has a pair. He has a native and an agricultural counterpart. And later on, we are also including Dutch agricultural soils. So this is actually the layout of the experiment that he designed, okay? So we have the whole, you know, the sampling sites here growing from north to south. We have two soil types. And then what Dario did is he collected all these soils and he brought them into the greenhouse for a controlled bioassay so that, you know, all the soils are basically exposed to the same. We did, you know, the native agricultural soil. You can use wild, land races, and improved. So there's a whole pedigree of potato germplasm here. You can grow them in these soils and then profile the microbiome on the roots, which he did. And then you can basically trace changes in the diversity during potato domestication. And the other thing that he did is he exposed these plants when they are growing in these soils to a leaf pathogen, Phytophthora infestans, which is the late blight pathogen, which is for potato one of the major diseases. It's an oomycete disease on potato. Okay, so this is how the experiment looked like in the greenhouse. So we brought everything in the greenhouse, And what Dario did, he did not do the pedigree experiment yet, where we had all, but he had just one domesticated potato line, which he grew in all these soils. So we did do microbiome-associated phenotypes. So we screened for tolerance to Phytophthora. We did amplicon metagenome analysis. We established already a culture collection from, so real microbes. And then this is still to be done, the pedigree analysis, where we do wild relatives, land races, and modern ones. So the phenotyping. This is, so they were grown in native and agricultural soils, and then we exposed these plants to the late blight disease on the leaves. So the soils, and then and this is the phenotype. So this is the native soil. You see plants exposed to, they look pretty healthy. Here they are diseased. Same story here with some differences. Here they are a bit the same, and here they are also a bit the same or even a bit worse in the native soil. And then he plotted basically the disease of these one potato line for all the different sites here. Native, agricultural, and you see that for many pairs, the disease incidence of Phytophthora is higher in the agricultural part of the pair of soils that they sampled. Good. This is where we are. So what should we do next? So we have the soils. We are profiling the microbiome and the metagenome. We did the phenotyping, right? So how to proceed? What would be the next experiment that you would do? Yeah, we already did. We have it in collection. Then you go for? Okay, yeah, but the microbes that we have are from the roots, and Phytophthora is on the leaves. So we're not looking at a direct, interaction, but we're looking at an interaction from the roots to the shoot. Okay? So the direct interaction, I say, yes, we could, for example, find, but it will not, let's say, be mechanistically insightful for what we see in terms of the phenotype. Unless these microbes, they travel from the roots all the way to the leaves, and then but I doubt that's the case. So, check for endophytes. Yeah, that's a good idea. Yeah, yeah. Mechanism of ISR. Yeah, very good point. So, one of the things that we're doing is to isolate RNA from the plant tissue and see if certain transcripts that would point to an induced resistance response are present there. Yeah. But what would be the very first experiment that you should do? Think about the lecture, the introductory lecture that I gave. I think we had 4 questions. You remember the first question? The first question was, what is the relative importance of the microbiome for that phenotype? Did we already demonstrate with these experiments that the microbiome has any relevance for the phenotype or not? We did? No. Exactly. So think about that. So what should we do actually with this? So we have the soils, we have the germplasm, we have the bioassay. What experiment should we do first to answer this question?
We want to see for the relative abundance and differential abundance of this files as well as while and the...
Yeah, we can do it, but think more simple. So,
We have to see whether the microbiome, the amount of this microbes present, they're relating to the resistance phenotype.
exactly, and how do we do that?
So we check for the microbiome, like we'll inoculate the pathogen and we check in which soils the plants are resistant.
Yeah. Yeah. Think again about the lecture that Victor gave about Pasteur. What did he do? He pasteurized, right? So the very first experiment that answering the question is to do the experiment again, but then sterilize the soils so that the phenotype that you see is not related to some sort of like physical, chemical, whatever property of these native soils that basically triggers the induced resistance response, because we don't know that from these results. So we sort of assume that the microbes are important, but we don't know, right? Good, so Dario did that experiment. And when he, for 3 soils, and when he sterilizes the soils and do the experiments again, indeed, the phenotype that you see in this native soil, he loses. So I mean, many people skip this. They immediately go to microbes, isolate, assuming that, and you know, and he even did the calculations with this GEM model, which I showed you earlier. And if by heart, but you know, don't sort of like record me on this, that it's, but I think the first calculation suggested that the contribution of the microbiome to this phenotype was around 11%. So that's super important to know. There is another experiment, so the sterilization is one, but the other experiment that you could do to show that the microbiome has an importance, what would be the experiment? Transplant, excellent. Yeah, so what he also did, Dario, he took 10% of the native soil and he transplanted that into the agricultural soil, which was gamma sterilized. And then indeed, he could confer the phenotype. Yeah? So these transplants, I think, you know, in plant microbiome, we use too little in terms of experimental tool. Because, you know, you can use it to reduce the complexity of your microbiome, which is associated with that phenotype. So sometimes, you know, it's all fancy with all these next generation, but, you know, think about the more, you know, sort of like mechanistic, simple experiments to show the contribution, right? Good, okay. So sterilization worked, transplant worked. What should we do next? We have already the markers to show that there is an induced resistance response. What should we do next? Good point. Yes, excellent. Yeah. So if you do a pedigree microbiome, you can look at the core microbiome. Again, look at the phenotypes, try to associate it with the phenotypes. Excellent. And then we have the core microbiome. We could even do some network analysis to see which, you know, genera or taxa are, you know, sort of like keystone hub taxa in the network. And then what would be next? Yeah. Excellent, but I think there's one step in between. So you identify the core based on your microbiome metagenome analysis, right? And then we need to go to the collection, right? The culture collection, try to identify these microbes, and then basically reconstitute the phenotype with the microbes that you see with the whole soil community, right? That's quite an elaborate type of analysis.
All the microbes cannot be cultured, so side by side we can go for the culture independent.
Right. Yes. Yeah. So you will run indeed trying to isolate, you know, the members of the core microbiome. You can run indeed into, you know, difficulties trying to culture those. Some of those. Yeah. That's what you probably have to deal with. Yeah. Again, this paper which I mentioned this morning, this high throughput culturomics, I think they claim that they could reconstitute around 70 or 80% of the microbes from this gut sample. But again, don't point me on the numbers. Have a look at the paper. But that's what because I just had a very quick, diagonal read through. So indeed, and then indeed what you said. So the culture, you have then the culture collection, and then from a subset or single or subset of microbes, you can then reconstitute the phenotype, and then for those ones, you could indeed, try to identify the mechanisms, right? One other thing. So, what organisms should we focus on? Because we are sort of like in this pipeline of focusing on the bacteria, but is that actually the right choice? Yeah, you can also go for it, but if you go back to the very original experiments that you do, right, with the sterilization and the transplant, what could you do to pinpoint which group of organisms that you could focus on? We also talked about that. The perturbation. So you could, if the transplant gives the phenotype, you could also treat the transplant with a fungicide or a selective antibiotic, right? And that already sort of like points you towards the groups of microbes that you could focus on. Yeah, again, that is not, it is quite crude, but it can point you towards bacteria or fungi, or maybe even something else, right? Okay, is that sort of like good for the mindset? Yeah? Okay, good. I will tell Dario, okay? Now, good. The last example I'm going to give you is the work that we do on domestication of tomatoes. And there we go a bit further also into, the plant genetic loci and even the molecules here. So, and the approach we did exactly the same type of experiments that I just showed you for potato. But we also had a very nice tool. We had a so-called recombinant inbred line. And a recombinant inbred line, that's a cross between the wild relative and the modern tomato. And the siblings there, after many generations, they contain, genetic fragments from one of the both parents, of course, and the ratio between those is different. And they profiled genetically, in every sibling of the recombinant inbred line population, which genes come from the wild ones and which ones come from the domesticated one. Yeah. And this is how the experiment looks like. It's the recombinant inbred line. We talk about 100 offspring that we tested. So you have the two parents, and then you have the offspring, which has genetics from either the wild or the modern one. So we grow them in, you can grow them in the wild, in the native soils, you can grow them in the agricultural soils, profile the microbiome, right? And the metagenome, that's what we did. 18S, 16S, so fungi and bacteria. So again here, this is the modern one, the wild one, Pimpinellifolium lycopersicum, and then there are the crosses, and then you have, a number of generations, and these seeds were then planted in the soil, and then roots, we focused on the rhizosphere, we profiled the microbiome and the metagenome, and then you get these sorts of, PCOA plots. I'm always looking at you when I present the PCOA plots. But you can see, very important here, that the wild and the modern, they separate. And then this whole cloud around it, that's basically the microbiome of the, the offspring of the two, of the cross. And then there is a tool which they call genome-wide association mapping, where you can associate, the genetics of the plant with a phenotype. And of course, the phenotype, many times they use these studies to have fruit quality or biomass or growth or development, all these types of phenotypic traits. But we use the microbiome as a phenotype of this cross, right? And for that, and you can look in the paper we developed, Ben Oyserman, Victor was also involved in this project. We developed some new bioinformatic pipelines to actually do this type of analysis. So they now call this mGWAS, microbiome GWAS, they call this now. And I'm just going to show you the end result of the associations, right? So you have, here you have the genetics of the recombinant inbred line population and the parental lines. And then here you have the diversity of your microbiome population with, yeah? And then what we found, and then you can look at significant associations between the genetics of the microbes and the genetics of the plant. And to my surprise, there were some really nice associations. So you find an association in water balance, which is trehalose, which is an osmoregulator in the microbiome, with genes or a gene clusters here involved in drought tolerance. Nutrient cycling came up, iron, phosphate, and then you can go through the whole list. And also exudates, we find a nice association between malic acid, came up again. You know, the example I gave yesterday with tomato, it popped up again in the analysis here with an association to the microbiome here, which was referred to as malic acid, utilization or degradation. And then, yeah, this is sort of like a broad map, but you can make from these types of studies, you can make these, what I call these metro maps, right? If you go to Paris or here, you go with the metro somewhere and there is a sort of like, a crossing point where you can choose, okay, I go to this place, etc. So this is how it looks like. So here it says these are the chromosomes of the plant, the tomato plant, and then here we have the microbiome at the genus level and at the class level and even phylum level here. And I'm just going to give you an example because there's quite some information, but it's still quite crude. So the regions, for example, on the chromosomes of the tomato are quite big. They can even go up to 28 megabases, right? So that's large. And then you start looking into these regions, these what they call QTLs, quantitative trait loci, and then you can pick a number of these genes that are there, yeah? And the risk there is that you go cherry picking, like, this is interesting. That associates with that, so it must be this. Okay. We did that, actually. That's human nature. You go into that type of cherry picking. So FIT here, that's an iron transcriptional regulator here. And aquaporin, that's one of the genes in the plant that is related to drought tolerance. That sort of like took our attention. And then you can see this one is nicely mapped to, a number of traits, genes in Streptomyces associated with this trait. Again, associated. And when we looked at the gene clusters in Streptomyces that were associated with, for example, the FIT and the aquaporin, there were desferrioxamine and coalicillin, okay? These are two siderophores of Streptomyces, so iron acquisition systems in the microbe. So we could sort of connect iron acquisition in the microbe with an iron transcriptional regulator in the tomato. Here there were some systems on sugar, import, and arabinose. And importantly here, actinobacteria were more abundant on the modern than on the wild. We've seen that also in the bean, the story. So anyway, so it's all bioinformatics, this. Of course, we started with an experiment, lots of data, and then we got these associations. Now, I come from, the time that, you gotta, get these microbes and do the real experiment, okay? So validation. Is this true, or is this just sort of like a computational dream, okay? So what Stalin Sarango Flores, PhD student on this project, did, he took the rhizosphere samples that we used for microbiome analysis from these tomato lines, and he started plating them on media which were semi-selective for streptomyces. So this is how these plates look like. Then he purified these streptomyces. They also have this mycelial growth. And then he started to sequence the genomes. It looks a lot like all the practicals that we are talking about here, right? But then the most important part was, are there streptomyces in our collection that at the genomic level, so comparative genomics, map to the streptomyces from the metagenome analysis? Because with the metagenome analysis, we also recreated the genome of this Streptomyces ASV5. Yeah, that was, and we were able to do that. So this is ASV5, and these are actually the isolates. You see the contigs here. They were not fully closed for several, but some of them were. And they are quite huge genomes, around 9.5 megabase. And then you can, and then we will go through that. You can do the annotation of the genomes. You can look at the traits that you also identified in the metagenome. Are they there in the isolate that you isolated? And yes, they are. And then what I always call the $1 million experiment, then you have to do the validation. So what we did is we took the streptomyces and we inoculated the streptomyces on modern and wild tomatoes, so the two parents. And remember, from the microbiome analysis, ASV5 preferred the modern tomato over the wild one. And fortunately, but you never know, indeed, so here you have the colonization, the density of the cells in a log unit. And this system here, this is not a tomato plant, but just as an illustration, we use the system, it's a transparent soil. So it's a sort of like, how do you call it? It's with alginate beads, which you can make yourself. And it resembles a bit sort of like the coarse structure of the soil, but you can look through it. So you can see the roots growing, and so if people want to have the lab recipe, I can provide you. You can easily make, just in the lab yourself. Transparent soil. If you want to do some imaging of your microbes on these plant roots, super nice. If you want to do chemistry on the roots, super nice, yeah? So ASV5, the isolate that we have, when we bring it back onto the plants, it indeed preferred to colonize the modern one over the wild one. And what is interesting, we also did phenotyping. And then when you inoculate the streptomyces, you could actually see that indeed plants inoculated with the ASV5 that's shown here, they have a higher biomass, both in the wild and also in the modern tomato line. But in the modern one, the increase in biomass was higher than in the wild one. Then you remember the system that I showed you yesterday with this below-ground olfactometer system? Yeah, where you can profile root volatiles. So Muhammad, he took those two parental lines, Pimpinellifolium and Lycopersicum moneymaker, and he profiled the volatiles on those two roots. And he could nicely show that for several of these compounds, there is a clear difference between the wild and the modern ones. And what we are doing now, so we have microbes, we have the compounds, and we're now trying to see, can we connect those two? So if you have a volatile more present in the modern one, is the streptomyces, for example, more attracted to that volatile than, yeah? So that's where we are. So this is also work in progress. So this was a bit about the studies that we did. Several years ago, Juan Perez, the PhD student on the bean project, he did a meta-analysis of other plant species. So he took all the raw data from the database and he tried to recreate, again, wild, modern. Can we find specific patterns that not only go for bean, but go for many plant species? And these were the studies that he used. So we worked on barley, on sugar beet here, There were some other plant species as well. And I'm just going to give you the end result. The overall picture was that Bacteroidetes, they preferably like the wild relatives, whereas Actinobacteria and Proteobacteria, they love the domesticated cultivars. I have to say, I think we need to redo that analysis again, because there's many more studies now where they have compared wild and domesticated. So to see if this is going to hold, right? But this was the cartoon that Juan was drawing. So here you have the plant phenotype. So the wild one here with a higher level of Bacteroidetes, and then the Actinobacteria and Proteobacteria on the modern one. Changes in root architecture, exudation, and also, the native soil here with a low pH in the native soils, and, more phosphorus and nitrogen and water supply in the agricultural soil. So this is basically bringing it all together. I think I should stop here. Where are we? Okay, good. Good. So again, lots of it is work in progress, but I hope I've given you a flavor of how you can use domestication and also plant genetics, plant breeding, to look at microbiomes. Are there any questions or suggestions for other experiments? Please tell me. Go ahead, Ramakrishna.
You have tried with the wild plants and domesticated ones. Have you tried any experiment with the hybrids?
Yeah, very good question. Good. Hybrids, super interesting. So there are some interesting studies where people look at two parental lines and the hybrid, and that is called the heterosis effect, right? So many times when you do that, you see that the hybrid of the two performs better than the two parentals, right? There is some nice work done in the US on that with maize, but there is also some other plant species. With the potatoes, we managed to team up with a company that is able now to grow potato from true seeds. And they also have created hybrids. And also there we see a heterosis effect, just simply like looking at the phenotype. What we have already done, Dario, he has done experiments where he also works in the native and the agricultural soil, and in the natural, but also the gamma sterilized. And what we see, and again, that's sort of like, there are many details, but the overall picture there is we see that the heterosis effect is also partially explained by the microbiome. So anyway, but very good question. Yeah, go ahead. You need a microphone? Trees, yeah. Good question. To be honest, I have no experience with perennials. It requires quite a different type of experimental approach to sample, right? And even to do the experiments. Yeah. Perennials for a PhD project, that's quite challenging, I should say, Are you working on perennials? Yeah. Yeah, I think you could do a lot of similar things with perennials, although, you got to have a bit of a long breath to especially with phenotyping. Absolutely, yeah, very good point, yeah. I have to think about this, yeah, the perennials. Yeah. Okay, thank you.
Sir, you were mentioning about the wild one and from where the microbiomes and they represent after domestication the microbiomes. Sir, I heard of that the microbiome that is with the wild plants, it has its own nature. For example, you were mentioning about its acidic soil.
Yeah.
The soil condition. If you would like to, for example, if transplanting those microbiomes to the what are the factors that will be affecting for the growth and survival of those microbes in the
Yeah, so that's a very good question. You know, in terms of transplants, you can first do phenotyping, and then what was suggested here, then you can go to isolation of the microbes, right, that are part of this core microbiome. And then with the isolates, the individual isolates that represent these, you could do sort of like the studies that you propose, like, so if you transplant them to an agricultural soil, are they really going to, you know, establish, survive, and do what they need to do? Because it's a very good point what you're making. Because indeed, you know, if they are not there in the domesticated ones and they are there in the wild relatives, they will probably have a hard time establishing on the modern ones, which means that on the genetics of your modern plants, you need to do something, you know, to be able to make them also sort of like competent and happy. Happy is not a good word, but, you know, on this modern one. And that's where probably the plant breeding comes in. And that's why, you know, we are looking especially into the exudates, you know, because that could be also quite an easy marker in breeding to screen for germplasm of modern ones that do have that, you know, specific or group of compounds that are needed to support the microbes from the native soils. Very good question. Yeah, I forgot to mention that.
Like we know that plants have been originated millions of years ago.
Here.
So we know that plants have been originated millions of years ago. And like you said, the missing microbes. So don't you think that from millions of years ago, from the wild relatives also the microbes have been gone missing?
Whether they have evolved, yeah, over this trajectory, yeah. Yes, I think so. I'm pretty sure, but to my knowledge, in relation to the wild and the domesticated ones, there is very little knowledge on that yet. Yeah.
Sir, and one more thing that during the domestication process, you said that there is a species gain. So don't you think that in the wild varieties also there will be species gain?
Well, I did not say there is a species gain, but that is one of the ideas that people have. Yeah, so you can either have a species loss, that's the missing microbes. Then there is a sort of like a species replacement. So in terms of diversity, things don't change, but you get sort of like a different composition. And then there is this last option where in the modern varieties, there is a species gain, right? Now, whether that's really true, we don't know yet.
Because of cultivation practices, cultural practices, for example, application of fertilizer and other things, that has promoted to specific.
Maybe you can use the microphone. Yeah, sorry.
That is maybe because of the cultural practices. For example,
Yeah, sure.
fertilizer application and other practices, which has made to gain with some microbes, for example, the actinum, what you are saying.
For sure. Yeah. And that's a bit of the complexity of the system which we're trying to separate, right? Okay, so what happens, you know, simply based on plant genetics, and what is linked to management practices, you know, and you got to separate those two. And that's why we do the experiments where we grow all the same plant species or the whole germplasm in all these different soils separately. Yeah, and then you can do the cross comparison to actually answer these questions. So we're just at the beginning, to be honest, to think about this. Good question.
So the question is, sir, in case of dysbiosis in the microbiome, wherein the soil has been saturated with like pathogenic microbes and all. So how can we go for the reclamation of those things with the use of these microbiome, transplanting or something?
Yeah. Also very good question. So the dysbiosis you're referring to, and the presence.
Because especially in case of scenarios where we have a limited land, especially small scale farmers or something. So they're repeated monoculture of some crops.
Exactly.
These kind of crops.
And you get a build-up of the
Yes,
pathogens there. Yeah.
Because this is a problem in case of perennials, especially if we talk about apple. Because there is problem of replant disease.
Absolutely.
People are trying to replant, but it's it's not establishing because the dysbiosis is so much.
It's there.
In
Yeah,
the soil.
exactly. Yeah.
The plants are not establishing.
Yes, yeah, that is, that's if you go to the domesticated, and management practices, that is one of the other points that sort of like complicates this whole type of analysis. Yeah, good point. So the answer to that, I don't know. The only thing what we did is there was one experiment where we looked at the impact of native agricultural soils and then gamma sterilized, and the microbiome contribution was to insect resistance on the tomato. Now, what was interesting from that experiment is that the domesticated tomato, so we took an insect which is endemic in South America, which is called Prodiplosis. And when we exposed the tomato plants, wild and domesticated, to the insect above ground, we could see that the domesticated plants were resistant. So apparently the breeding apparently introduced unintentionally also resistance to this Prodiplosis. So, and then when we gamma sterilized the native or the agricultural soil, the domesticated plant was simply resistant. So it did not rely on the microbiome for resistance to the pathogen. That was just an inherent genetic trait. However, when we do the wild tomato ones, so we grow the wild tomatoes in the native and the agricultural, in presence of the microbiome, it was protected from the insect. If we gamma sterilize those two soils, native and agricultural, it was lost. The wild tomato became sensitive to this insect. So which told us that the wild one relies much more than the domesticated on the microbiome for protection against this insect. So in terms of dysbiosis, that was your question, and the build-up due to, yeah, it would be super interesting to, develop some experiments into that direction to, yeah, deal with this issue here, yeah. But an interesting experiment in your case would be, is to use some, soil or microbes from either wild apple varieties or, and then transplant this into these, dysbiotic replant disease issues, and then see if the microbiome from the wild and the native soils could have a beneficial effect there. Yeah. Yeah, you're welcome. I hope that I also, and again, have an eye for it. With the lectures, sometimes you see these plots, you see so all the things that you learn, with the computer practicals, you see it back in the literature every time. So they do a differential abundance analysis. They do genomic comparisons, try to cross-link your microbial isolates with the microbiome analysis. So I hope also these patterns are clear from the background literature. Good.
From the wild to domestication, you also mentioned that bacteriates were more in the wild case, and Actinobacteria and Proteobacteria are more.
Yeah.
I have a doubt that, sir, if we want to, for example, phytophthora infestants, which you were mentioning for its resistance, where the infestation was more in the cultivable, indoor crops. So if we want to bring the microbes, if they are affecting on the disease resistant part, so if they are giving any resistance to that disease, and if we want to bring that to the cultivable form.
Yeah.
So is there any chances of replacing any bacteria or the microbes of the wild one affecting the current bacterial microbes or microbiome of the current cultivator plant, and that would in turn affecting plant
yeah, that is well possible. Yeah, absolutely. So if you introduce a microbe into the system, and if you do that early on, thinking about priority effects, you will displace other microbes that are normally in that system. Yeah. Keep in mind that in the modern system, when you would bring a microbe from the native or the wild relative to the modern one, you are doing that because the current situation of the modern one is a dysbiotic condition, right, which you want to change. But there are a lot of studies where, and I think here in India it's the same, when you want to use microbes as an application, you usually have to provide a risk assessment analysis, indeed addressing these types of questions. So if you introduce a microbe, how persistent will that microbe be in your agricultural system? Which organisms does it displace? Is that displacement, is it temporarily, or is it sort of like permanent, right? So all these questions that belongs to a risk assessment type analysis. And that's an important component of the whole registration and stuff. So yes, absolutely.
Because in our current, even without knowledge or without knowing or unknowing, there are many good things, good microbiomes in the current domesticated plants also. So that way also we can't replace them because that might affect.
Yeah, so super. Yeah, you already know what I'm going to talk about on Monday, I think. So Monday, what I will do, in my lecture, in the keynote lecture, I will talk about do we really need to introduce microbes, or can we make use of the functional potential that is already there in the indigenous population. And I will give an example from the work that we do in Ethiopia, a very practical way how to make this happen. So stay tuned.
Are microbiomes specific to a group of plants? Like if we are isolating a microbiome from solanaceous plants, example tomato or brinjal, and if we are introducing the same into cereals like rice or any other crop, so will it have the same effect as it is so the origin?
Yeah, good question. So there are a number of studies where they have compared the microbiomes of not so much, genotypes within a given plant species, but where they have compared multiple plant species growing in exactly the same soil microbiome pool. And what you see there is that you get also at the plant species level, you get sort of like quite a separation between microbes that prefer to grow on cereals or wheat, and even within cereals. So there's a huge differentiation. I mean, you can always do the test, of course. Yeah, if you have an interesting microbial consortium, if you have interesting microbial isolates, which originate from, let's say tomato, of course you can also test them on a number of other plant species for a particular phenotype. Sometimes you can be lucky in terms of that it does, establish, and it does give a beautiful phenotype. There are many examples of that. And you can see, for example, that many of the microbial inoculant products that are, provided by companies, those microbes originate, of course, from one particular source. And then the companies, they start testing them on other crops, and sometimes, it works, sometimes it doesn't. And that has to do with the specificity of the communication between the plant and the microbe. Yeah. Go ahead.
On Thymosphere, we could see that the same microbial genus and species, they do exist in monoparts and dicots. We have already the data sets.
Yeah.
We see the Hydrea as much as homogenic and also in rice. I'm sure it must be in wheat as well. So the homogeneity exists in the canopy in terms of the abiotic factors. I would say that there may be a common microbiome on the canopy. Rhizosphere, maybe the scenario may be different.
Yeah. But again, I want to come back to it all depends on the purpose that you are. So for example, if your target is to find microbes that, for example, control pre-emergence damping off or post-emergence damping off. So there's a very short time frame of the developmental of the crop that the microbe needs to do what it needs to do. Then, of course, being persistent or being super colonizer of these on the longer term is less relevant, right? Yeah.
Sir, like we are using and that is going to alter the phenotype of a plant. Will that phenotype will be transferred to its offsprings too, sir?
Will the phenotype be?
That will be transferred, or it will be like it will be added to its offsprings.
To the offspring. Oof.
Vertical transmission of the phenotype
Yeah.
is induced in the parent by the microbiome.
Yeah. There are some groups working on epigenetics where they look at whether, you know, when you apply a microbiome to a plant, if that sort of like beneficial effect, actually, so if you then harvest the seeds from that plant and you grow them again in a new system without the microbes, do you then have a residual effect in the next generation? There are some groups working on that. And then there is an epigenetic sort of like basis of that. I'm not familiar yet with success stories there, but I know that there are some groups that are working hard on that. Yeah, that would be even fantastic, of course, because then you have one, you know, application at one point, and then you can go into the generations. Yeah, very good.
So here means what is the basis of strain selection? Or we have only randomly selected the wild relatives and the domesticated one? Because the microbiome, it will be different for different ecological zones. So
Yeah, so with plants, of course, you know, a very important basis of selection, as I indicated also in my lecture yesterday, is the chemistry, the underlying chemistry of the root exudation. That's a very important. And root exudation is not a simple trait. So if you grow plants in a physical, chemically different background, you will get a difference in exudation patterns. It will differ in terms of developmental stages. It will even differ among, you know, sort of like the localization along the root axis where, you know, which exudates are released somewhere. There's some beautiful reviews on that even show the spatial changes in root exudation pattern. So also in terms of your ecosystems that you're working with, you will find, you know, a different basis of selection. Yeah, so when you do these types of assays that we do, you know, there is actually, you cannot have enough germplasm, you cannot have enough soil types, you know, to try to find these general patterns. The 9 whatever germplasms that we used for bean, we found some beautiful associations, etc. But in fact, in retrospect, when I think about it, you know, we could have tested so much more germplasm to find even more fine patterns in those data. The tomato one with the 100, well, that's a good start, I think. But even that could be expanded more, yeah. But there's only so much you can do within, yeah, okay. Okay, yeah, thanks. This was super nice. Thank you for the questions also. That gave another dimension. What shall we do, Kumar, in terms of the time? Shall we go with the next lecture, or are we going to go for a coffee, or what's the story?
I will break for tea now.
Excellent, good, yeah. Okay.